Good morning, everyone. Welcome to the ISTH-sponsored Gene Therapy in Hemophilia Symposium. My name is Dr. Pantep Angchaisuksiri from Bangkok, Thailand. I am an APSTH and ISTH Council member and chair of the ISTH Education Committee. I will be chairing this symposium today. Gene therapy offers the potential for a cure for patients with hemophilia by establishing continuous endogenous expression of factor VIII or factor IX following transfer of a functional gene to repress the patient's own defective gene. Great progress has been made in the development of gene therapy for hemophilia in the last decade. Today, we have three world experts in gene therapy in hemophilia who will update us on this exciting topic. Dr. Flora Peyvandi, she is a professor of internal medicine at the University of Milan and the director of the Angelo Bianchi Bonomi Hemophilia and Thrombosis Center in Milan, Italy. She's also president-elect of the ISTH. Dr. Stephen Pipe. Dr. Pipe is a professor of pediatrics and pathology at the University of Michigan, Ann Arbor, USA. He's also the medical director of the Pediatric Hemophilia and Coagulation Disorders Program and the Special Coagulation Laboratory. Dr. Alok Srivastava is a professor in the Department of Hematology and head of the Center for Stem Cell Research at the Christian Medical College of Alok in India. The objectives for today's symposium are to outline the current and emerging approaches for treating hemophilia, including various approaches of gene therapy, to identify key characteristics of current clinical trials in gene therapy for both hemophilia A and hemophilia B, to identify and critically evaluate important safety and efficacy in clinical trial outcomes under consideration for gene therapy as a treatment for hemophilia, and to recognize key concerns and unknowns related to the future of gene therapy for hemophilia. This activity has been planned and implemented in accordance with the accreditation requirements and policies of the ACCME through the joint providership of the France Foundation and the ISTH. The France Foundation designates this activity for a maximum of 1 MA-PRA Category 1 credit. Here are the disclosure policy. Here are disclosures of the faculties and staff. Disclosure of unlabeled use is shown here. This symposium is supported by educational grants from Biomarine, Freeline Therapeutics, Pfizer, Shire, Spark Therapeutics, and Unicure. The agenda for today is as follows. Overview of gene therapy for treatment of hemophilia, which will be presented by Dr. Steve Pipe. Updates on clinical trials of gene therapy in hemophilia A and hemophilia B by Dr. Flora Peyvandi, and other evolving initiatives in gene therapy trials in hemophilia by Dr. Alok Srivastava. And we will have a panel discussion by all faculty at the end. To claim credit, please use the link shown on the slide. So without further ado, please welcome the first speaker.
Hello, I'm Stephen Pipe from the University of Michigan, and I'm pleased to give this overview of gene therapy for the treatment of hemophilia. We've seen some incredible series of advances in the past few decades that have really only accelerated in the last few years. The replacement therapy era began in the late 1960s, early 1970s with plasma-derived clotting factor concentrates. And persons with hemophilia were one of the earliest to benefit from the recombinant DNA era. And this technology has been leveraged further as targeted engineering has further improved the biochemical properties of these recombinant molecules. We're now in the early experience of the non-replacement therapy era with a humanized bispecific antibody, emicizumab, which substitutes for factor VIII replacement. However, other antagonists of the natural physiologic anticoagulants remain under investigation, specifically small interfering RNA knockdown of antithrombin, monoclonal antibody inhibitors directed against tissue factor pathway inhibitor, and a bioengineered SERPIN that targets activated protein C. Now, we're focusing today on investigational gene therapy. And now, although gene therapy includes gene editing and cellular therapies and can involve either viral or non-viral vectors, the first clinically available technology will be through gene addition using viral vectors. What is the rationale for gene therapy? Well, this figure depicts the typical pattern of factor levels achieved with exogenous clotting factor concentrate replacement therapy in the lower half of this figure, and then a resultant hemostatic effect as depicted in the upper half of the figure. Standard half-life factor concentrates maintain minimally effective trough levels with a regimen that would be typically every other day dosing, for example, with factor VIII. The extended half-life therapies can reduce the frequency of administration burden and also maintain higher trough levels. Both of these strategies with factor replacement result in the achievement of zero bleeding in only a minority of patients, with breakthrough bleeds in prospective trials that range from 1 to 6 bleeds per year. The non-factor therapies change the treatment paradigm by providing a steady-state hemostatic effect, but without a measurable factor level. The steady-state hemostasis is likely a key contributor to some of the enhanced efficacy observed with the non-factor therapies as compared to traditional factor replacement. But the promise of gene therapy is to achieve a steady-state, measurable level of factor that's above key thresholds that would also produce a steady-state hemostatic effect that would be sufficient to prevent bleeds following a single treatment intervention. The first wave of gene therapies for hemophilia are adeno-associated virus vector-based liver-directed approaches in which a functional copy of the factor VIII or the factor IX gene is packaged inside of a viral vector that's been stripped of its propagating viral gene elements. And then following a single outpatient IV infusion over as little as 1 to 3 hours, these particles are picked up by liver cell receptors. They're taken up into the cell. There, the vector particle uncoats and delivers the DNA to the nucleus of the cell. There, the genetic elements that accompany the gene allow for efficient expression and ultimately secretion of the factor VIII or the factor IX protein into the plasma. Those factor IX and factor VIII elements ultimately reach a steady state between secretion and clearance, and that's represented by a factor level that we can measure with traditional blood sampling. There are some hurdles here, however. Many people will have encountered versions of these non-pathogenic viruses previously, and they may have specific antibodies that would block their uptake into the liver. For 25 to maybe 40% of patients, they would be excluded from this therapy on account of this. The immune system also gets another crack at responding to this viral vector because after uncoating within the cell, the protein elements of the capsid will be broken down, and these can be presented on the surface of the cell to adaptive immune cells. And if these cells react to what would be seen as a foreign protein, an immune response that destroys the cell could ensue, taking with it any potential factor expression. Now, this has been manifest in the trials by elevations in common liver enzymes that must be diligently followed post-infusion for weeks and months afterwards. And if signs of toxicity appear, this will trigger an immunosuppression course, typically with oral corticosteroids, and there is the potential for some additional drug-related toxicity. So despite these hurdles, the studies that you're going to see today will show that this works in the great majority of individuals, and the immunosuppression course can either be avoided or is short-lived for many. So if we're talking about gene therapy as a new type of drug, what are the elements of this drug? So Some of the components here are the therapeutic transgene, that would be factor 8 or factor 9. The transgene promoter, these are the genetic elements that are going to drive the expression of factor 8 and factor 9, but in a tissue-specific manner, for instance, just within the liver. You need the transgene delivery system, in which case here we're using adeno-associated virus vectors as the delivery vehicle. We're targeting an appropriate host cell. In this case, it's the hepatocyte. And then we have protein outcome metrics. So what are the clinical outcomes of the patient having expression of that factor 8 or factor 9 protein, both laboratory as well as clinically based? We're talking about viral vectors for gene transfer. Currently, these are the most efficient means of transgene delivery. Adenovirus have been used. Retroviruses include gamma retroviruses and lentiviruses, and then the adeno-associated virus that I've just mentioned. All of these have been evaluated in hemophilia, but the AAV vectors have shown the best safety and efficacy balance to bring these to the clinic and advance them towards pivotal trials. AAV vectors are considered the delivery tool of choice for in vivo gene therapy. This is because they're non-pathogenic. They can transduce dividing and non-dividing cells. However, a limitation of the AAV vectors is the packaging size of the expression cassette, which is limited to a transgene component of about 5 kilobases of DNA. So the two components of this viral vector is the transgene cDNA construct and then the adeno-associated virus itself. So we mentioned that the limitations for the transgene are up to about 5 kilobases in order to fit inside of an AAV capsid. These transgenes are typically they typically undergo codon optimization, in which the nucleotide sequence is optimized for human codon reading, but it doesn't alter the amino acid sequence of the transgene. There is promoter selection and optimization to try to improve expression and tissue specificity. And many of these transgenes may include some upstream introns and enhancers to optimize that expression further. With respect to the adeno-associated virus genome, these are non-pathogenic human parvoviruses. They have little innate immunogenicity themselves. But there's many serotypes, and those are driven by variances in the capsid protein, the outer packaging of the AAV. The vector DNA typically forms stable extrachromosomal episomes, circular DNA if you like, which can then form concatemers of multiple copies within the cell nucleus. We think a very small percentage of the DNA integrates into the chromosomal DNA of the host. Now, the wild type AAV genes are depicted here with these inverted terminal repeats and then the rep and cap genes. But when we're talking about converting an AAV virus genome into an AAV vector genome, we remove these viral rep and cap genes and we replace it with the therapeutic transgene and its upstream enhancer promoter elements. And as we've discussed, these tend to be liver specific. The factor 9 cDNA as the therapeutic transgene is easily packaged within the AAV. The full-length factor 8 is far too large to put into the AAV vector. So we are limited to B-domain deleted forms of factor 8, and of course these are used clinically. But even then, the cDNA is right at the upper limits at approximately 5 kilobases. So for AAV factor 8 vectors, these vectors are actually overstuffed in order to fit the transgene into these. It's these ITR elements that help facilitate the episome formation. So AAV permits in vivo protein production, but there are many variables at an interpersonal level which can contribute to some of the variability, unpredictability, and perhaps even lack of durability that we've seen in the clinical trials. If we look at the pathway from the IV infusion of the AAV vector to hepatocyte protein secretion, a lot of this biology is actually poorly understood. Some of the steps where we believe there could be contributions to some of the variability and unpredictability include where the AAV binds and then is internalized at the cell surface receptors, how the AAV escapes from the endosomes before those endosomes go to the lysosomes for degradation. What are the variables that control nuclear entry and decapsidization of the AAV vector? Some of the efficiencies with which the single-stranded DNA is converted into a double-stranded DNA. Some of the contributions to how efficiently the episomes and concatenators form, elements of variability in mRNA transcription, and then variabilities related to translation and post-translational processing. On top of all of this, we can look at each person's liver as a approximately 1.5 liter human bioreactor. But this is unique to that individual. They're not identical. And this is in contrast to some of the stainless steel fermenters that can be up to 20,000 liters that are used for recombinant protein production. So gene therapy in hemophilia is becoming an exciting choice in the field of hemophilia treatment. The clinical trials, as you see, have demonstrated that one single intravenous infusion of an AAV vector containing factor VIII or factor IX cDNA can achieve high protein expression levels in many patients. It can achieve long-term durable factor expression, particularly for those receiving factor IX transgenes. For most participants, we see an absence of spontaneous or traumatic bleeds, and the vast majority of patients are able to cease our ongoing requirement for prophylaxis. Now, what are some of the efficacy considerations in gene therapy for hemophilia? These can be divided into the physiologic and clinical parameters, as well as some overall mental health aspects of health-related quality of life. Within the physiologic and clinical realm, we're going to be looking at the frequency of bleeding events post-gene therapy, what factor activity levels have been achieved at steady state, what's the duration of expression, and improvements in, say, pain and discomfort as it relates to bleeding or chronic joint problems. On the mental health side, we want to be able to capture what this does to the patient's overall emotional functioning after they've been transformed by this kind of therapy. With regard to safety, the adverse events can be grouped as short-term, long-term, and then mortality events. And within the short-term adverse events, we're looking for any evidence of liver toxicity, inhibitor development against the transgene, the immune responses to the gene therapy that I introduced you to, and also we're concerned about whether this triggers thrombotic events. With regards to long-term adverse events, we want to be paying attention to development of any other disorders related to having gone through gene therapy, increased attention to vector integration into the host genome, and then we want to have some further understanding of the duration of vector neutralizing responses, as this has implications for whether a patient could receive any type of this type of gene therapy in the future. We recognize that the remaining unknowns over the long term after gene therapy will require that we follow patients well beyond the follow-up of the sponsored trials. And so to this end, the ISTH organized a multi-stakeholder group to establish core outcome measures for data collection. These have been incorporated into the World Federation of Hemophilia Gene Therapy Registry. This manuscript is now published. And by following patients globally over long periods of time, we will hopefully be able to get answers as to the key determinants of durability, reliability, variability, safety, and factor activity levels. Now, let's hear from my colleagues as they review some of the outcomes observed in the clinical trials to date.
Good morning. Good afternoon, good evening. I'm Flora Peyvandi from University of Milan. I want to thank the organizer and also ISTH for this invitation, and also France Foundation for helping us to organize this educational program on gene therapy. My role would be to update you on clinical trial of gene therapy in hemophilia A and B. For this audience, I think it's obvious that hemophilia is caused by deficiency of factor 8 or factor 9, and that's due to the gene mutation, which is creating a chronically debilitating disease due to bleeding episode affecting soft tissue, joints, and muscles. Lots has been done in the last 50 years trying to prevent the bleeding, moving from replacement therapy, standard product, to longer acting product, and now we are having the facility of treatment by subcutaneous infusion once weekly or even once monthly. And then we are moving in the future to gene therapy, which is actually on the clinical trial. And my role is to bring your attention to where we are standing with the gene therapy. Gene therapy is replacement of a dysfunctional gene by exogenous functional gene in order to cure the disease, or at least to have the expression of the protein for a long period of time. What you do need for making the gene therapy to work, you do need to have a fragment of DNA or gene, in this case factor 8 or factor 9, which is inserted into a vector. The vector could be adeno-associated, adenovirus, retrovirus, lenti, or even non-viral. Once this gene is encapsulated in this vector, should be pointed to a target tissue. Then the vector releases the gene into this target tissue by binding to the receptor and trying to make a long-term expression. Adeno-associated vectors are those that has been used for treatment in hemophilia until now, and they are considered one of the best delivery tools in vivo. They are not pathogenic and can transduce both dividing and non-dividing cells. There is a limitation of the usage of adeno-associated in terms of packaging the size of the expression cassette, and approximately that is 5 kilobase of DNA. And that's the reason why they could be easily used for factor 9 gene therapy, and for factor 8, we do need to make the modification of the cDNA and make it suitable for this type of vectors. In the gene therapy, it's very important the pre-existing immunity against the vector. The serotype of the vectors are having different prevalence of immunogenicity in the general population. And usually AAV5 is the one with the lowest immunogenicity in the general population. And that data has been reported by Biomarine in 2019, followed by AAV8, AAV6, and AAV2 is the one with the highest immunogenicity. But if AAV5 is the best, why we are not only using that vector? And that's because you do need also to understand which vector is suitable for what type of tissue expression and what are the tissue that you are targeting for. Let's go to see the results of gene therapy in hemophilia B. Fortunately, we have important results since almost 10 years ago, when Professor Amit Nathwani and Ted Tottenham at University College of London started to treat the first patient with gene therapy and having the escalation of the dosage achieving 8 to 10% of activity. However, the further modification in the construct of factor 9 improved significantly the efficacy of treatment, and the expression by introduction of Padua variants jumped significantly. And right now, we have two phase 3 clinical trials, one Unicure, and the second one is the Pfizer or Spark. And we have also phase 1 and 2 clinical trials, which are very active and ongoing. Let's go to see how are the results. The phase 2b data on 3 patients in AMT-061 of Unicure showed that using 2 or 10 to 13 particles or of the vector is creating an interesting level of expression by a mean of 31% at week 6, 41 after a year, and 44% almost after 2 years. And the additional important data is that all patients had neutralizing antibodies against AAV5. And that's the first time that we are seeing a result like that. And the presence of neutralizing antibodies is not reducing the level of efficacy and expression. And it's important to understand also how is the variability. And as you can see, there is a really good level of inter and interact variability of this clinical trial with almost constant expression. So as the efficacy, there is no bleed post-treatment, no requirement for factor 9 concentrate. And as a safety, none of the patient experienced elevation in transaminase, and no participant required steroid treatment, and there was also no inhibitor development. And if you look on the data of phase 3, at 1 year on 54 patients who received a single dose and completing 26 weeks of follow-up, you can see that 23 out of 54 patients did have neutralizing antibody against AAV5 at baseline. There was no correlation of pre-existing antibodies with factor 9, and post-treatment level of factor 9 was 37.2% at 26 weeks. About 98% successfully discontinued their routine prophylaxis, and also treated bleeds have been reduced by 91%. I think there is no doubt there was a good level of efficacy and safety. 9 steroids for increase of transaminases have been reported, 7 infusion-related reaction, and actually that was 10% of the dose that has been infused. And the highest titer of the antibody was reported to be 1 to 3,000 as a dosage. Unfortunately, we have seen an unfortunate result regarding one of the patients who was treated with AMT-061, which showed a preliminary diagnosis of hepatocellular carcinoma. This patient had previously different risk factors associated with HCC, including 25 years of hepatitis C, hepatitis B, and non-alcoholic fatty liver disease and advanced age. Due to all these reasons, it's becoming very difficult to understand any correlation between the gene therapy and the clinical situation of the patient. Both the company and the regulators now, they are looking accurately on the data to understand whether there could be any relation between the gene therapy and the development of hepatocarcinoma. Let's go to look on the durability of hemophilia B gene therapy. As I explained before, the results of St. Jude's and the UCL, which is about 10 years ago, but the last data on published data is after 8 years, which was reporting the level of expression using the wild type factor 9 was between 2 to 5%. Followed to that, we have Unicure that I showed before, AMT06. We have the expression of almost 4 years using the wild type about 7%, followed by the Unicure with the Padua mutation for 2 years, which is bringing the results of 41%. And we have also Spark with factor 9 Padua gene mutation reporting the result between 9 to 47%. Here you can see the level of variability are higher to what we have seen in the Unicure data. What about hemophilia A? As I showed before, with Adeno-Associated Vectors is a little bit more difficult, factor 8, because of its size and also because of its immunogenicity. So that's the reason the results that we have achieved for factor 8 gene therapy in the last years is amazing, and how fast we have achieved these results. Right now, we have 3 clinical trials on phase 3. The most advanced is Biomarine 270, which has used AAV5 Adeno-Associated Vectors, and I'm followed by Roche and Sangamo results. And there are also other additional clinical trials ongoing in phase 1 and 2. Unfortunately, 2 of the trials are also opening for hemophilia A patients with inhibitor. I think it is important to look on the data after 4 years of dosing of Biomarine, and there is inter-subject and intra-subject variability. At 26 weeks after dosing, we have seen the highest amount of expression, and you can see that in some patients is more than 150%. There is a loss of factor 8 through year 4 at the lower dosage. And if you look, this is very gradually, and about 20% loss every year. 4 patients have been used to be treated with 4 to 10 to 13, and 7 patients with the higher dose who experienced a mild and non-serious and transient elevation of ALT and AST, and they have been treated with steroid. And it's very clear if you look on the data that the lowest dose lost almost the expression at year 4, and that's the reason why the company at phase 3 clinical trial went with the upper dose, and we're going to see the results later on. Mean factor 8 usage was reduced by 96%, and that was a very good data in terms of clinical efficacy, from more than 130 infusions to about 5 per year in both groups. Here is the results of phase 3, and a large group of patients have been treated in the clinical trial of BMN 270, with 112 patients receiving 1 single dose of the highest dosage that I showed before, and the level of expression after a year remained more than 40%, about 43%. Using chromogenic assay, that means with the 1 stage, we probably get 20 to 30% more level compared to chromogenic. And in a subset of 17 patients, those at 2 years, we can see still the expression is there, and we have the reduction up to 24.4%. Even here, we can see that the ABR was reduced about 84% with 99% reduction in consumption and the factor VIII infusion rate in treated patients. 86% of patients did show elevation of ALT, and corticosteroid was used to manage the ALT. And we look forward in the future to have more and more data from Biomarine in terms of understanding the efficacy of steroid and also the safety of long-term treatment of the product. No thrombotic event have been seen, and no participant developed inhibitor against factor VIII. Second clinical trial of hemophilia A in phase 1 and 2, which is a SPARK, is a modified AAV that carries a bioengineered factor VIII expression cassette. 4 patients with less than 1% and no antibody against AAV have been treated with a lower dosage of the vector, and activity of factor VIII was in range of 6% to 22% at 1 year or 52 weeks. 3 patients did stop prophylaxis, and 98% had a reduction of annualized infusion rate, and 85% of them had ABR, which was reduced after a follow-up between 15 to 18 months. 1 patient with ABR of 18 before vector infusion reached the 0 ABR. There was no serious adverse event even here. No patient did demonstrate increased level of ALT and AST, no inhibitor development, and 3 patients did receive a steroid cycle after 3 to 6 weeks post-infusion. And these preliminary results showed an increase in factor VIII activity and also the safety of a SPARK trial at 1 year after the first dose, which was based on 5 to 10 to 11 as an amount of the vector. And here again, we can see the durability of the human gene therapy for hemophilia A, and Biomarine, as I said, is the one with the longest period of study, which is becoming now almost 4 years, with a level of expression up to 10% to 35, with this variability from phase 1 and 2, and the level of expression at 1 year is going to be 43% in phase 3, and the SPARK therapeutic after 2 years with the expression between 4 to 22, and Sangamo with an interesting expression of 57%, which we look forward to have that evaluation in the next step. As regards the adverse event, I tried to put together the data that we have available, bringing here, and you can see that elevation in liver in transaminase is the main toxicity, intermediate toxicity, and the majority of the events have been managed with steroid, and that's the reason I think we do need to make the combined data in the future, understanding the level of the steroid that is safe and its duration, and whether there is any beneficial effect upfront treating the patient. Some cases have been associated with a partial or complete loss of expression. And with this slide, I'm going to make the summary and bringing what is the issues that we need to think and where should be the focus of the science and research in the future. There is no doubt that clinical trial of gene therapy improved significantly. more than what we were expecting in the last 3-4 years, with a long-term therapeutic expression, much longer for factor 9, about 10 years, and 4 years remaining around 10% to 20% for factor 8. And established efficacy in decreasing bleeding and also the usage of product. No discussion about the efficacy. In terms of potential toxicity, we have recently seen in dog animal models after 10 years, there was integrating of gene vector and the trace of the vectors over there. However, none of these vectors showed to activate any oncogene or creating any type of hepatocarcinoma in the dog model. There are still unanswered questions: liver toxicity, duration, variability in response that in some clinical trials are bigger than the others, which require our attention. Optimal regimen of immunosuppression is not completely clear, and mainly the discrepancy between the factor assays, one stage or chromogenic, and as I said and showed, the biomarine result, for example, is efficacy shown by chromogenic, and for factor 9 might be different. And that's the reason why ISTH in future, through factor 8 and 9 committee, should focus on the standardization of the assay. And finally, I think for the intermediate and high and long-term safety, as has been established by the FDA and EMA, we do need to have a long-term gene therapy register. And this registry has been discussed between World Federation of Hemophilia and ISTH and all other patients' organization and scientific organization located in European country and also in United States. And there would be different stakeholders and the companies thinking together with regulators what are the best information that should be collected in a minimal data set or even maximal, depends on what we want to collect, and also for a long period of time all over where the patient is treated. With this one, I want to thank you, and I hope this lesson and educational program was useful for understanding gene therapy. Thank you.
Good morning, everyone. My name is Alok Srivastava, and I'm a hematologist at the Christian Medical College, Flora. It is a great pleasure to be part of this joint ISTH-APSTH session on gene therapy for hemophilia. I'd like to thank the organizers for this invitation to talk about initiatives outside of Western Europe and North America for developing gene therapy for hemophilia. What you may have known and heard reiterated in the previous two presentations are the science of gene therapy for hemophilia and the various programs for developing suitable products for providing gene therapy to patients with both hemophilia A and hemophilia B. These currently include the AAV vector and gene editing based technologies, which are already in the clinic, and the lentiviral based vector approaches, which are in development. This list, put together by the World Federation of Hemophilia, shows the current clinical trials using the AAV vector, more than 20 of them, at various phases of development, and also some of the lentiviral-based gene therapy efforts, the first of which is something that we are associated with. In fact, our efforts include two AAV products, one based on AAV3, which is a collaboration with the University of Florida and the Emory University in USA, and the other using AAV8, which is a collaboration with a pharmaceutical sponsor in India. I will, of course, tell you more about these in the later part of this presentation. What this collage shows you is some of the current gene-modified cell therapy products licensed by the EMA or FDA for a range of diseases with different levels of efficacy. What I would like you to note is the remarkable cost for each of these products, ranging from about $500,000 to more than $2 million or euros for each of them. While these costs may be challenging but surmountable in some of those countries, they will really be unthinkable in most other parts of the world. So, for if no other reason but for survival and for some form of equity in access to these transformational therapies, we need other efforts, and they are crucial. Talking about these programs is a little difficult because there's not so much published information, and what is available is not always up to date. Starting with Japan, we know that researchers there have a long and strong history of preclinical work towards gene therapy for hemophilia B in the non-human primate model using the AAV platform. They have also screened their population to look for seroprevalence of the anti-AAV antibodies. More recently, work has been initiated towards applying the CRISPR-based gene editing technologies for gene therapy for hemophilia. A clinical trial is also being planned in an academia industry partnership involving the GT Medical University and the Mitsubishi Tanabe Pharma. This effort has some challenges to overcome, but given the proactive approach of the Japanese regulators and industry towards cell therapy in general, we will hopefully see this moving forward soon. The next country to talk about is China, where again there is a long history of gene therapy research. In fact, China has one of the earliest efforts towards gene therapy for hemophilia in the early 1990s, but we do not have enough details of those studies or publications about them. More recently, there have been applications of gene editing technologies in certain controversial applications, but it did show the ability of those technologies for more suitable therapeutic applications. Currently, there are 2 registered clinical trials for hemophilia A and B in China, and at least one of them is recruiting subjects right now. We look forward to seeing presentations of its results. In Africa, the efforts are at Johannesburg in South Africa, with Dr. Johnny Malangu's group trying to set up collaborations with various sponsors to initiate clinical trials of gene therapy in that country. They have already gained experience by having some of their patients participate in Europe-based subject recruitment. They're also participating directly in one of the current gene therapy clinical trials. They have also initiated AAV antibody seroprevalence program to choose potentially eligible patients. In South America, most of the efforts are in Brazil with Dr. Margaret Ozello and other colleagues. The Brazilian hemophilia treatment centers are participating in some of the sponsored clinical trials shown here. A major issue to resolve, of course, will be the access to these therapies for patients in these countries once these products are approved. I'm sure suitable arrangements may have been discussed already, and we look forward to learning about them. In addition to these efforts, the St. Jude Children's Hospital from Memphis, USA, is also leading an effort to bring gene therapy to certain low and middle income countries as a no-cost access program. This is to be initiated as clinical trials, and the program is being developed. We look forward to seeing further details, as this is also being done in collaboration with the World Federation of Hemophilia. So that brings me now to some of our efforts in India. They started more than a decade ago with collaborations with colleagues at the University of Florida and the Emory University in Atlanta. There are two programs which I'm going to show you some details of, the preclinical work, which is ready for IND applications, and one close to approval for initiation of a phase 1 clinical trial. The first of these efforts is to develop an AAV3 vector-based product for gene therapy for hemophilia B. Here we have several innovations, starting with the AAV serotype, AAV3, which will be the first time this will be used. It then has a unique promoter. There are other modifications, including the codon optimization strategy, which is shown in the right cartoon, where compared to the LP1 that was used in the Amitnath Wani study, this actually shows double the expression. So we are hopeful that this vector, including the Padua variant of the factor 9 sequence, would give us significantly higher expression than what has been seen with the classic LP1 wild type factor 9 codon optimized vector in the past. This vector has been tested in two models, as shown in this slide. On the left, you see a hemophilia B mouse with the transgene packaged in AAV8, showing a comparison with the LP1 vector of comparable expression on the left and a dose-dependent increase in expression on the right. As you know, AAV3 does not infect mouse tissues, and therefore we had to use a humanized liver knock mouse to test the AAV3 vector with this transgene, and that's what's shown on the right cartoon. And what you find here again is that there is a dose-dependent response with this transgene, giving significant expression of factor 9. So this was completed some time ago. We are now in the process of establishing production of this vector at a facility in India. And as soon as that has been completed in the next few months, we will be filing an IND for an AAV3 based factor 9 gene therapy to be initiated in India. Now, you've heard a lot today about AAV based gene therapy for hemophilia, and there are some very important positives. You heard the ease of administration as an outpatient therapy. You've also heard that there are minimal safety signals that can be directed, there are minimal safety signals and almost no serious adverse events. You have also heard that there are minimal safety signals and no serious adverse events have been associated with AAV-based gene therapy. But there are also some major negatives. The important one is the presence of pre-existing antibodies. Now, this may be as low as 20 or 30% in some populations, but in many others, it is much higher than 50%. And some of our recent studies in India looking at pre-existing antibodies to AAV3, 5, and 8 have shown very high prevalence, a data that was recently published. The other major problem I see in using AAV-based therapy is that this is not yet been designed for use in young children less than 10 or 12 years of age, because we would like the liver to reach its adult mature size before administering AAV-based gene therapy, so that we have the full complement of hepatocytes that can be infected with these particles for the transduction to occur. If you treat much younger children, we do not know what the sustenance of that expression will be and whether the amount of factor produced will be adequate for later in life. So these two issues make it a real challenge, because if we have to offer prophylaxis for the first 10 or 12 years to these children, that's a task that has not been achieved and will then be associated with all the complications of hemophilia without adequate treatment. So we need to look at other options. And one of the options that we have looked at in collaboration with the Emory University colleagues is to study the lentiviral vector with a factor 8 transgene with a specific promoter that will allow its expression in the monocytic lineage. So this vector was evaluated again in two preclinical models, one using this transgene with the lentiviral vector in the hemophilia A mouse using the mouse hematopoietic stem cells, and the next using human hematopoietic stem cells in the NSG mouse. This data was published about 2 years ago, and what it showed was significant expression of factor VIII that was secreted in quantities that would make a major difference to the clinical phenotype of severe hemophilia A. Based on this data, an IND was filed in the US as well as in India. This IND actually has been approved in the US, and we have a longer regulatory review process in India at the moment because, if approved, this will be the first gene therapy trial in the country. We have gone through several reviews, and we are near the end of this review process, and hopefully we will be able to initiate this study soon. So what this product includes is the lentiviral vector, which then would be used to transduce autologous hematopoietic stem cells, and the product candidate will then be given as a single IV injection after conditioning of the patient, like for an autologous stem cell transplant. So certainly this is very different from an outpatient intravenous injection because this will be much more involved. But the advantage is that this could be offered to very young children once it's known to be safe and effective, and there would not be any ineligibility related to existence of pre-existing antibodies. So what this will involve is collection of autologous hematopoietic stem cells, which will then be exposed to the lentiviral vector with the factor VIII transgene. This processing will then give us the product, which will be tested to make sure that it has achieved the required release criteria. And with that, we will then be able to use this product for an autologous stem cell transplant. Now, this is certainly much more invasive than what has been done for gene therapy for hemophilia so far. But this is not new to the world because this approach has been used for immune deficiency disorders for more than 2 decades and is currently in use for several major hemoglobin disorders gene therapy. What we have done in the last 6 months, because the Indian regulator wanted us to establish these techniques in our lab in the country, we have actually collected peripheral blood stem cells from 3 patients with severe hemophilia A, done the transduction, and shown that we can achieve the required vector copy numbers to initiate the clinical trial. So this data is being submitted to the regulator to hopefully to receive their approval for initiating the phase 1 clinical trial. So with that, I'd like to thank the colleagues at the Center for Stem Cell Research and the Christian Medical College Vellore who are involved in supporting this trial, but also very importantly, colleagues at the Emory University, especially Dr. Trent Spencer, and at the University of Florida, Dr. Arun Srivastava, who have been long-term collaborators on this program that has evolved over the last decade. Of course, all of this requires funding, and none of this would be possible. Interestingly, this is all funded by the Department of Biotechnology of the Government of India. So to end, I'd like to mention that developing gene therapy, as you heard in the first talk, requires the technology, the vector, and the transgene that gives you the proof of concept data, its validation in preclinical models, and then clinical grade production of the product. But then after that, you do need regulatory processes in the country that are able to evaluate the product and the clinical trial proposals, and then suitable sites to conduct such studies. And finally, you need scale up of these operations because after the initial phase 1 or 2, you then need to make these products in quantities that can make them available to the large numbers of patients in the world. So we believe that we want to go on that path and try and offer options for those who may not be able to afford the current gene therapies. We would like to model some of this work on the efforts at the Fondazione Telethon at the San Raffaele Institute in Milan. They have a long history of research in gene therapy, trying to develop several products, which are now reaching the market in collaboration with various pharmaceuticals. The important thing is to ensure that these products are affordable and accessible to those who need it most. And with that, I'd like to stop and remind you of this quotation from William James Mayo. The aim of medicine is to prevent disease and prolong life. The ideal of medicine is to eliminate the need of a physician. We hope that one day, with safe and effective gene therapy, we will be able to achieve that for people with hemophilia. Thank you.
So the last part of this symposium is the panel discussion on what does success look like. I have some questions for the speakers. Flora, in terms of efficacy, are you more focused on factor expression levels or evidence that breeding events have been substantially reduced or eliminated as the key indicator of success?
Good question, Pantep, which is not really easy to answer. I think at this point, at this stage, we should consider both outcomes important. The level of expression associated to the clinical manifestation of the patient. We do need to understand what is the level of expression. Without that, we cannot understand how much our system is working. But it's also very important to understand the type of the assay that we are using. And that could be only helped by the clinical manifestation of the patient. And especially when the level of expression is getting lower than 20%, that's really fundamental and is a key point.
So I do agree that we have to look at both factor levels and clinical breeding after gene therapy. Yeah. But assuming that the success is based on factor expression, what stable level of expression is needed to consider gene therapy a success?
That depends on which price you paid for the gene therapy. In terms of the clinical efficacy, we have seen, I think, if you have more than 20% or 15%, you do not have spontaneous and you might prevent also arthropathies. But of course, you are not protected from traumatic event. And if you can justify in the future without having excessive problem of toxicity, then you can increase the level of expression. But if the level of toxicity is getting higher and higher, I think you should find the level of expression which is suitable for the for prevention of the bleeding, and I think that should be 15 to 20%.
Yeah, because I think the concern if we give too much vectors to the patient might cause toxicity, especially transaminitis, correct?
Actually, both, to be honest, because we have this mid-term toxicity or short-term, how you want to define it, as you said, the liver toxicity, which we did not understand 100% its relationship with the amount and type of the capsid. But of course, that's an important issue. And the second point is a long-term, because it's true that we have seen the integration is about 8 to 10%, and the number of the virus that are getting vectors that are getting to the liver anyhow is not really very low. So if you are increasing the amount of the vector, you are increasing also the risk of the vectors that getting integrated. I think all these points should be considered in the future. So more efficient vector with a less amount of that, it makes much happier, I think, the patients and the physician.
Mm-hmm. So you're happy that the level doesn't have to be normal or near normal, right? So just 15 or 20% should prevent spontaneous bleed.
Yeah, I think, there is a really different type of vision in the patient's opinion and physician. I think until we do not have really good data on the safety, I think we should go scale by scale and not pointing out too much.
So this question is, given the uncertainty surrounding the efficacy of subsequent gene therapy infusions, what is the minimum duration of stable factor expression above a protective level needed to justify the risk and cost of gene therapy for hemophilia?
More or less what we achieve right now, as I showed, for factor 9 is about 10 years, and there was a good level of expression because that was a wild type of factor 9. And for factor 8 is about 4 years, and still the expression is remaining around 20%. So I do believe that we're going to get the 5%, at least about 10% of activity. So that's more or less what scientifically we see. Now, if there would be any integration, and that level of integration is responsible for a long term, more than 5 years, then we have to understand what is this level of integration and which level of expression is remaining. But right now, I think we can think about 10 years, about 40% of factor 9 and 10%, I hope 15% for 5 years.
Mm-hmm. So how long should we follow the patient after gene therapy?
I think, to be honest, the patient should be followed up all their life, maybe with less intensity, and the intervals could be a little bit larger, but still we do not have to lose the patient and their trace, because the data that we have are very limited and we need to understand more and more. But minimum 10 years, I think.
Mm-hmm. So to date, gene therapy for hemophilia using AAV vectors have been relatively safe and tolerable in clinical trials, but the number of treated patients is low. When can patients be confident in the long-term tolerability and safety of gene therapy?
I think we need to change a little bit our vision of looking to data only in the world of hemophilia. And we have to look on all patients treated with Adeno-Associated for the different type of diseases. And if you put all these patients treated together, so maybe numbers are getting more and the power of the results would be stronger. However, as you said, for example, as you've seen with the Biomarine data, the regulator asked for 2 years at least data to understand more than 122 patients treated in order to have a stronger data. But I think once we have that data, we can just join it to the rest of the gene therapy in order to understand more.
Mm-hmm. Yeah, I think human long-term safety data is still limited, but reassuring. But I think more data are needed. I mean, the last question is, what are the main barriers to gene therapy for our patients with hemophilia at the moment, in your opinion?
Yeah, I think there are different barriers right now, to be honest. I think the most important thing is education. And I don't know if you remember, we have done actually an exercise with the ISTH, and we went through this survey to understand how physicians, they feel confident with the gene therapy, and there were about 50% they were not quite feeling comfortable about gene therapy. So that's first. We need to really teach to our patients and mainly to our physicians what is gene therapy, the cost and organization of the centers for gene therapy, because gene therapy is not like factor 8 and factor 9. And for the dosing of gene therapy, you do need to have lots of facility. And it's like a multi-domain task to put different experts together, like infectivologists, virologists, and different people to understanding. If everything goes well, that's fine. But if something goes wrong, there should be people, expert lab, expert physician, that they work together and trying to solve any complication. So creating a good network of treatment in terms of how one spoke as is now starting to be planned with the EHAD and EHC in terms of the European Organization for the Gene Therapy.
Yeah, I do agree that education is very important for both healthcare providers and the patients, and also for the payer. That's why we have this symposium today. So I think we are out of time. And before I close, I would like to thank everyone for participating in this symposium. Thank you.